The Institute for the Study of Peak States presents the 4th International Psycho-immunology and Psychobiology Research Symposium. In today's presentation, Psycho-immunology versus Psychobiology, which one is better for treating diseases, by Dr. Grant McFetridge.
Okay, and our final speaker for today is our founder at the Institute, Dr. Grant McFetridge. So I introduced earlier that he originally worked in electrical engineering R&D as a consultant and was an adjunct faculty in California. In 1998, he founded the Institute, where he is focused on research. He is the author of Subcellular Psychobiology Diagnosis Handbook, Silence the Voices, Peak States of Consciousness, Volumes 1 and 2, and the Wholehearted Healing Manual. So Grant is here to present psychobiology versus psychoimmunology, which is better for treating diseases. Over to you, Grant.
Thank you, Shane. All right. Well, as Shane said, my name's Grant McFetridge, and it's been interesting listening to the talks today because I don't always know what people are going to be actually saying, and it's very interesting to have heard how much of the talks boiled around exceptional states of consciousness, particularly beauty way. And the reason is because now that we have working psychoimmunology treatments where we can actually get rid of pathogens, we can make progress on that core project. So, but I'm going to move back a little bit and just talk about more practical things for therapists who are actually interested in what we're doing on the nuts and bolts. Okay, so let's see. So I'm a practical person who cares what you call it. Psychobiology, psychoimmunology, what the hell is all this? So I'm going to go back to the basics for some people, but I think it'll be useful. What are we covering today? Well, we're covering what developmental and subcellular psychobiology are, and what the heck is psychoimmunology, and how do they differ? And finally, how do I use them? What are they good for? And you can go back to sleep for those of you who are in the back of the room. Okay, so this is a boilerplate slide that I always throw in about safety and liability. Ask yourself, are you a Darwin Award nominee? The stuff we're talking about here is for educational purposes, to keep the public informed about what the heck we're doing. We don't want you to be going, I want to experiment on myself and not have a clue about what the hell I'm doing. Okay. If you want to read about safety, we actually threw up a training video that we use in-house on our main website, peakstates.com, safety.html. Okay, so what exactly is psychobiology? Well, if you look at our website, we have peak states, psychobiology, and psychoimmunology as major headings. So what is psychobiology? Well, it comes in two flavors: developmental psychobiology and subcellular psychobiology. Okay, so I threw up the textbook definition of psychobiology, and I'll just read it to you. Psychobiology describes the relationship between psychological and behavioral processes and the underlying psychological mechanisms. Okay, or you can also say psychology uses biological perspectives to describe and explain psychological phenomenon. It's got a lot of different names in the field: biopsychology, biological psychology, physiological psychology, and behavioral neurosciences. Okay, so that's kind of the field. Now, how do we define it? Well, we agree with those previous definitions, but we're more hands-on in the way we do stuff. So when we talk about psychobiology, we're often looking at markers. What are emotional, experiential, or kinesthetic markers for disease or disorder or problem that a person has? We use those markers both to figure out what's the causes if we are in research, but we also, for clients, we use the markers for both diagnosis. If they have the marker, that means we have a treatment for it. And how's their treatment going? Did the marker go away? And a great example is Asperger's syndrome, which has an amazing marker where it feels like the person's embedded in a glass test tube. And they can actually experience this themselves if they're guided to how to sense it, and they can monitor their own progress. Okay, so in psychobiology, you could say that the real key to psychobiology is knowing what questions to ask. And that's the tricky bit. Okay, so what is developmental psychobiology? Well, you could think of it as a bit of a subcategory of general psychobiology. And it has to do with this organisms, including men, and how prenatal, perinatal, and early child development affected them. Now, this is incredibly important for the Institute because we discovered so much of the issues that we wanted to solve happened before we're born, either during the birthing event, the implantation event, the conception event, or some other random event in prenatal development. Really, really important. So developmental psychobiology is key to what we do. Turns out it's also key for peak states. And as some of the speakers have already mentioned, wholehearted healing was developed back in the 90s, primarily to allow us to study these prenatal events. At the time, there really were no good ways to get into prenatal trauma reliably and easily. And Wes did a great talk about that earlier today, as he described those early forays into prenatal development. Okay, so what's subcellular psychobiology? That's another flavor of this psychobiology, and that was something we discovered back in 2002. And it's not in the field of psychology yet, mostly because we've been applying it and using it and developing it, and haven't really taken the effort to try and push it into the world. But basically, it has a model, what we call the primary cell model, which says that our experience of life is made up of two things. It's our normal experience, where we have a body in the world and we stub our toe and it hurts. And another experience where our consciousness is actually inside one particular cell of the body. And it turns out that one particular cell is incredibly important because that's where you could say we live and we guide the rest of our being. So those two environments, the outer body and that one primary cell, superimpose experience together. Now, why is that important? Well, because it says that if we screw around with that primary cell and we fix something, that echoes out into all cells in the body. Now, there's some exceptions to that, but generally that's an absolutely true statement. Okay. I should probably mention some of the exceptions have to do with cancer. Okay, so psychobiology and actually developmental psychobiology used together really are amazingly useful for figuring out diseases of unknown etiology. Okay, so what does that mean? That means there's a lot of diseases out there that people are clueless about, like Parkinson's disease. Millions, many millions of dollars poured into trying to figure out what it is, so hopefully they can figure out how to fix it. Diabetes research, another endless money sink. Just a lot of different diseases that no one knows what the underlying mechanisms are. And what's beautiful about subcellular psychobiology in combination with developmental psychobiology is that we can look at what the heck's going wrong in the primary cell and say, aha, that's the cause of the disease. And as our slide shows, generally it's directly or indirectly caused by pathogens. So I love the little example of hearing voices is caused by a particular fungus. And some diseases are caused by these, indirectly caused by these pathogen interactions. Again, the Asperger's disease, good example of that. And some problems that we see in people are actually caused by mechanical damage in the primary cell. And I like the example of scintillating scotoma. I think I said that correctly. That's where your eyes see this like bright, scintillating crack. And I had this happen to me recently. We tracked it down to damage to a particular organelle inside of the primary cell. Now, interestingly, that organelle didn't just break, which it appeared to from having this scintillating crack in my eye vision, but it was actually caused by a particular prion disease that was interacting with that organelle and damaging it. Okay, so again, this is all review, but for some people, I hope this is useful. If you've not seen this work before, what we found is trauma itself is based on something inside the primary cell, biological. And in this particular case, it has to do with inhibited gene expression. So just briefly, when you need to do something, you need a protein to do it with. And the map for proteins is DNA. So something happens, you need to do something, and you what's called express the gene and make an RNA copy of it. Now, normally that RNA copy, it's a little bit of string of nucleotides, goes out into the cytoplasm, gets read like ticker tape, and you make a protein. It turns out that trauma is due to the fact that there's, you could say, damaged histone, or I like to think of it as crap on the surface of the gene, and the RNA duplication process gets buggered. So it gets partly out, and everything stops. And that's actually the physical basis for trauma. As you might imagine, we spent a lot of time figuring out very cool ways to fix those stuck genes. And it turns out that a lot of trauma, actually all trauma healing techniques, or most, I should say, interact with this stuck structure in the primary cell to get their effects. Okay. Oh, I should add this little comment on the left. This isn't just hypothetical. What's really been exciting for me in the last few years is researchers have found really very interesting experiments with models like nematodes and such that show that the RNA is actually carrying the trauma information when they take it out of one organism and inject it in another. Okay, so in our view, trauma therapies are a subset of psychobiology, subcellular psychobiology techniques. And they work by changing the interior of the primary cell, albeit maybe in just a minor way in the case of one particular gene. And generational, biographical, and associational trauma types all are dependent on different places inside the primary cell and how genes express. So that's pretty exciting to just be able to understand this emotional trauma upset stuff and to turn it into, hey, it's biology. And it's a biology we can interact with in ways that no one ever imagined before, because they're all caught up in the drama, or did you get hit, or, you know, were you kidnapped or in a hostage situation or whatever. Okay. So that was psychobiology. And I'm 15 minutes through, so let's see. What is psychoimmunology? Well, this, as a field, a scientific field, started when I believe his name was Adler, was experimenting with rats, and he found that he could affect their immune systems after he basically associated it with an immune suppressant and a harmless taste. And later, when he fed the harmless taste to the rats, they suppressed their immune system. And what really caught his attention was it was killing a bunch of the rats. And all he'd given them was basically sugar water. So I believe it was sugar water. But the point was, holy shit, there's a real interaction between making associations in these rats and actually affecting their immune system. So that was 50 years ago. And in the alternative community, there's tons of interest in, okay, how do our emotions affect our health? So you could say they're sort of in the same ballpark. Okay. Okay, so the conventional definition of psychoimmunology, you can go ahead and read the slide. But it says that what psychological factors affect the immune system so we can make the patient either immune or resistant, okay? And it's also called psychoimmunology, neuroimmunology, and it goes from there. They keep adding syllables to this stuff. In our work, we basically just call it psychoimmunology because we figured out all this stuff based on the primary cell, not at the brain. And since it's in the primary cell, let's keep it simple. Psycho, as in trauma healing, and immunology. Let's get rid of this damn pathogen. Okay. So I remember back in the 80s and 90s, this was really an exciting field, but it basically crashed by the 2000s because nobody was making any progress in actually how to implement these hopes and dreams. And I have to say, it's been a miracle after it's 37 years that we've actually made progress and succeeded in solving this problem. Okay, so how did we succeed in solving this problem? Again, this is review. We have the medical model of disease where we've got a castle and the pathogens, be it a virus or a bacteria or whatever, are trying to smash down the walls of our immune system. And that's pretty much what you get in school and pretty much everybody believes. The problem with that is, how do you fix this problem? Well, you try and take pills to induce a stronger immune system and blah, blah, blah. But what if that model is just flat out wrong? This was our model, and we call it, you could call it self-sabotage model. I like to call it the Trojan horse model. And that's where the pathogen comes up like someone saying, I have some sweet candy. Won't you just take me into your house? And that's what people do, even when the pathogen is going to end up killing them. So essentially, our body is tricked into pulling in the pathogens and hanging on to them. The good news is, with this model, you can do something about it, because you can say, well, what stupid reason do we have for wanting to pull this in? We can identify that reason and get rid of it. And boom, the pathogen is ripped to bits. And if you have the ability to see inside your own primary cell, it's really quite exciting to watch these things just completely fragment into parts and dissolve in seconds. Okay. So again, we solved the PI problem in 2020, and we now know how to use trauma techniques to target specific pathogens of whatever species. It's a little more than that. We have to take research time to actually figure out how to do a particular trauma, but it's now to the point where, yes, you do this step 1, step 2, step 3, and we've got it down to basically a real technology. Okay, the healing is typically very fast, usually 1 to 3 office visits, and what's exciting about this is its symptoms are fast. So if you did the job correctly, poof, the symptoms are gone. And it is kind of shocking sometimes to see these people who have been devastated by disease for most of their life, and boom, the symptoms just basically disappear right there in front of your eyes. Okay, so that's the good news. Now, so up to now, I've been doing a review, and now let's get into which is better. Do we want to use psychobiology techniques, or do we want to use psychoimmunology techniques? Well, it turns out it depends. Now, why does it depend? Well, the devil's in the details, as they say. There are trade-offs. Okay, so what do pathogens do to us? Well, as I wrote here, they're nasty. They give off toxins, they steal food, they eat our membranes, they screw with us to modify the cell interior to make themselves more comfortable or to reproduce, so on and so forth. A great example of that is type 1 diabetes. Worse than that, pathogens influence us. Not only do they do the Trojan horse thing where we want them, but some species are psychoactive and they can control our behavior, kind of a Stockholm syndrome. So the Borg fungus, which gives us the experience of culture, I'm an American, or I'm a Canadian, or I'm a Russian, or I'm a Israeli, or whatever, and we feel different from others who are outside of our culture. That's due to this particular fungus. And finally, this is even kind of stranger. Normal people want to use the pathogen inside their cells. So there this organism is, it's Trojan horsed in, and now we say, oh, well, I have a use for a Trojan horse. What if I have this horrible kind of feeling? What if I stuffed a Trojan horse over there? I love the example of pneumonia bacteria. We originally discovered that one of our colleagues' cats had pneumonia. It was in the vet's office and got really, really sick. And we realized it was lonely. And shortly after, the owner, I believe it was the owner, no, it was another one of my colleagues, got the pneumonia bacteria, and same thing. They were lonely, and they wanted that bacteria to make themselves feel less lonely. Okay. We also use it in other ways. For example, we might use the bacteria to patch a hole in a torn membrane and so on and so forth. So there's two problems here. We've got the Trojan horse problem, and then the fact is we also want to use them for stuff to make ourselves better, feel better. Okay, so let's boil down to how we look at pathogen psychobiology techniques. Well, we do two basic approaches. One is we treat a pathogen one little bug at a time, or one little bacteria at a time, or whatever we're doing. It's causing us problems, and we interact with that particular individual pathogen to get rid of the problem. Sometimes we make the pathogen healthier so it doesn't bug us so much. And our textbooks are full of that particular approach because it was kind of like the low-hanging fruit. We could actually figure those out first. Now, the next category, which I'm calling the pathogen species approach for psychobiology, is a new breakthrough in the last year. And what we realized, which had been a hypothesis we had for nearly 20 years, but we couldn't implement it. Recently, we've actually verified that we can find a trauma that makes us want to interact with the pathogen. And if we heal that particular trauma, boom, we don't want to interact with it anymore. We're still infected, but now instead of having it all over the place doing crap to our primary cell, we can do this little process, and it moves off into its own little corner of our primary cell and just sits there. That doesn't get rid of the pathogen, but it gets rid of most of the symptoms. Sometimes SUDS goes to 1.5, something like that. And for someone who's really suffering, it's pretty close to being completely well. They go, whoa, this really worked. And it's really simple. So both approaches work. The one is what we've had for 20 years, and you can buy it in our textbooks. The new one is still in the research, but it's amazing. And both approaches are valid and used in different circumstances. Okay. For example, it could be a lot faster to use the old approach for a particular client issue, or it might be better to use the new approach, which involves trauma healing. Okay. So, yeah, just more on the same. Psychobiology techniques can repair damage to the primary cell. We can use trauma healing to actually repair stuff, unless, of course, there's an aggressive toxin that's, as in the case of my eye, that's screwing things up. As I mentioned, these special cases that we have in our textbooks are basically psychobiology approaches. And we have a whole book on that sort of stuff called the Subcellular Psychobiology Diagnosis Handbook. Getting down here on the wire. Okay, Sam, what does the psychoimmunology approach have as advantages? Well, there's some pathogens that are just bad news, and you do not want them even stuck in a corner. Maybe they're filled with a particular toxin, or they're doing bad things to your cell in other ways. You just want them out of there. I'd say probably a good example of that would be type 1 diabetes, because it wants to screw with your cell and modify it to suit itself. Also, as Dr. Lykkegaard has just mentioned, some peak states like Beauty Way are blocked by pathogens. You need that pathogen out of there to make your Beauty Way totally stable and to even get it in the first place. Okay, so the psychoimmunology approach has a next problem, and as Dr. Lykkegaard alluded to, we resist giving up our pathogens. As silly as this sounds, not only do they cause us problems, but we don't want to let them go. It's like they're little toys or teddy bears or a connection to mother or you name it. We just don't want to let them go. And as Dr. Kirsten mentioned, it took us almost 2 years to crack that problem, how to solve it so people don't want to hang on to their pathogens anymore. So psychoimmunology approach now has basically 2 steps. One, getting rid of our resistance to letting go, and then actually healing the stuff that makes us let it go. Okay. So here's a kind of a cool table in the few minutes that I have left. The far left column, the psychobiology single column, which I guess I can point to right here, is you can go ahead and read this table, but essentially it's what we've been doing in our textbooks. The next one over is this sort of species level psychobiology approach. And we've coined a new word to describe that. We call it's psychogen healing. Bit of a story behind that, but just accept it for now. So, it's pretty cool. And so of the 2, it sort of depends on the circumstances, which one you would use, okay? The final one is psychoimmunology, and psychoimmunology takes just more steps. But when you're done, your SUDS is flat gone. It's 0. There's no chance of having that problem come back or cause you toxin issues, residual toxin issues, or anything of that sort. Love the PI approach. It's just more steps. It's a bit more work. So if you look at this and you think about clients, some clients are going to respond well to healing, say, trauma about sadness. They're not going to do well, say, on a trauma around anger. People vary about what they're willing to face and what they're willing to do. And so with this table, you can often figure out with some overlap which one you need to do, and can you get your job done with, say, a simpler process or a more difficult process. So there's trade-offs that the therapist would have to evaluate on the fly with a particular client. Okay, so how well does it work in practice? Psychoimmunology is pretty fast, 1 to 3 sessions, and you're pretty much done. And then we throw in what we call rule of 3. We make sure that they didn't miss something in their healing, that it's really finished. And we're doing a ton of work on different diseases, isolating exactly which traumas they need to be healed with and how to do it easily and so on and so forth, making sure it's safe. That's what we're up to now. We also have pay for results. When we start releasing this to the clinics, which we hope to do fairly soon on different diseases, it's always pay for results. That means if it didn't work, it's free. Or if it was unstable, it's free. So as you can see, the research team really has to do a very tip-top job to make this work properly. Finally, safety. Well, that single pathogen stuff is pretty well understood. We've been doing it for decades with thousands of people. The newer stuff is relatively new, from 1 to 3 years, and it's still being very carefully tested and in small scale. Again, you can look on how we do safety testing on our website. Okay, in summary, with no minutes left, what can you take away from this? First of all, our primary cell is like a bloody sewer. People say, well, can't you just fix my problem? Well, think of digging into an outhouse and trying to find a particular poo that you need to get rid of. There's a lot of crap in there. And so when we do our work, we have to be very careful that we're not interacting with other problems, that everything works out properly. Okay, symptoms. Some of those symptoms are endemic. In other words, as Mary was saying earlier, Kirsten was saying also, we just think it's normal to have these symptoms and that people act this way normally. And so sometimes it takes us a while to figure out what the symptoms are coming from? What's causing those? And finally, chronic diseases. What's pretty exciting to me is, in our experience, pretty much all of the chronic diseases we've been able to figure out so far are caused by different primary cell pathogens. Okay. Let's see, we talked about trauma therapy. Now let's get to the fun stuff, as Mary was going on about. It's really a revolution in medicine. So, and not only that, it fits right in with conventional medicine. That's why we work with MDs and cell biologists and drug researchers so well, because this stuff is hardcore biology science. And what's pretty cool is it can also be used to figure out new drugs. The big problem in drug development is, what the hell's causing the problem? You know, a lot of times with the work that we're doing, we can often point out the pathogen that's causing the issue, and other options for treatment can be developed using conventional techniques. Okay, if you want, there's a bunch of references. We have a bunch of textbooks, and I don't think we have time for questions. Shane?
We always have time for you to answer questions, Grant.
Well, you know, maybe we should just roll into the next one if you think the questions are.
Let me see if there's some that are going to be specific for you.
Okay.
So Evan's asking a question is, what is the origin of these parasites? You know, are they natural? Makes no sense that Gaia evolved these.
Well, that's a great question. I've often wondered about it myself. As a practical matter, we just take it as it comes. But it's a wonderful question, and I'm hoping that one of the graduate students in years to come will figure it out for me. Next question.
I think a lot of the other questions could be answered by multiple people. So maybe we'll stop here and then we'll get set up for the panel presentation. Thanks,
That's
Grant.
great. Bye-bye, everyone.
Thank you for watching. For more information, please visit www.peakstates.com.